So hello guys, my name is Jeffy Santos and I'm going to tackle the eukaryotic cell structures, and I hope you enjoy it. Okay, let's begin. Eukaryotic cells are defined as a cell containing organized nucleus and organelles which are enveloped by membrane bound organelles. So, example of eukaryotic cells are plants, fungi, and by the way, the eukaryotic cell, the EU is true, while the karyo refers to a nut or a nucleus. Are so named because they have a true nucleus in that their DNA is enclosed by nuclear membrane. Okay, by their genetic material is organized in chromosomes. The cell membrane, centriole, cytosol, lysosome, raw endoplasmic reticulum, the short term ER, vesicle, ribosomes, um, peroxisome, flagella, cilia, microvilli, mitochondrion, Golgi apparatus, and nuclear membrane, the nucleus and nucleolus, and smooth endoplasmic reticulum are parts of eukaryotic cell. So, what are the definitions of their structures of the eukaryotic cell? Okay, here we go. The cell is enclosed and held intact by the cell membrane, which is also referred to as the plasma, cytoplasmic, or the circular membrane. Cell membrane is like a skin around the cell, separating the contents of the cell from the outside. So, cell membrane regulates the passage of nutrients, waste products, and secretion into and out of the cell. Cell membrane is similar in the structure and function to all of the other membrane that are found in eukaryotic cell. Nucleus or nuclei that controls the functions of the entire cell and can be thought of as the command center of the cell. So while the nucleoplasm is the really gelations of matrix or base material of the nucleus that chromosomes are embedded or suspended in the nucleoplasm. The nuclear membrane, the nuclear membrane that around the nucleus is contains pulse or nuclear pulse, okay, with the word pulse. Okay. So through which the large molecules can enter and exit the nucleus. Genes are located along the DNA molecules, described as a band of string, and Each gene contains the genetic genetic material that enabled the cell produce a gene product. So, while the endoplasmic reticulum, or yeah, well, I said earlier in short term is DR, is highly convoluted system of membrane that is interconnected and arranged to form a transport network of tubules and flattened sacs within the cytoplasm. Okay, designated as a raw endoplasmic reticulum. This raw appearance is caused by the many ribosomes attached to the outer surface of the membranes. So, endoplasmic reticulum, to which some are not attached, is called smooth endoplasmic reticulum. So, the Golgi apparatus. Golgi apparatus is also known as a Golgi body that connects or communicate with the ER. Membranous sacs completes the transformation of newly synthesized proteins into mature, functional ones and package them into small. Membrane includes vesicles for storage within the cell or export outside the cell. The Golgi complex are sometimes referred to as a packaging plants. So the mitochondria.
The energy necessary for a circular function is provided by the formation of high energy phosphate molecules such as the adenosine triphosphate, or what's called the ATP. ATP is energy carrying or energy storing molecules within the cell. Flagella. Flagella possess relatively a long, thin structures are called flagella. To be flattened or motile flagellated protozoa are called flagellates. The swimming motion of the flagella enables the flattened flagellated cell to swim through liquid environment. Flagella are said to be web-like, while the cilia are also organelles of locomotion, but they tend to be shorter, more hair-like, thinner, and more numerous than flagella.
So we're finished to tackle the eukaryotic. And now I'm going to tackle the prokaryotic cell and their structures. So let's begin. Prokaryotes are single-celled organisms that earliest and the most primitive forms of life on Earth. As organized in the three-domain system, the prokaryotes include the bacteria or archaea. Some prokaryotes, such as cyanobacteria, photosynthetic organisms, are capable of the photosynthesis. The prokaryotic cells are about 10 times smaller than the eukaryotic cell, so this is too small. So structurally, Prokaryotes are very simple cells when compared with the eukaryotic cells, and yet they are able to perform the necessary process of life. Many prokaryotics are streamophiles, and can live and thrive in various type of stream. Environments include hydrothermal vents, hot spring, um, swamps, wetlands, and guts of human and animals. Prokaryotic bacteria can be found almost anywhere, yeah, anywhere, and are part of the human microbiota. And they live on your skin, yeah, in your body, our body, and every object in your environment. So, the bacteria cell anatomy and external structures are pilus. So, plasma membrane, what you call that? The nucleoid, cytoplasm, ribosomes, and flagellum. So, the prokaryotic cells are not as complex as a eukaryotic cell. They have no true nucleus, as the DNA is not contained within a membrane or separated from the rest of the cell, but is called up in a region of the cytoplasm called the nucleoid. So, prokaryotic organisms have varying cell shapes. The most common bacteria shapes are spherical, yeah, rod-shaped, and spiral. Okay, using bacterial as our simple prokaryote. The following structure and organelles can be found in the bacterial cells. So here it is. The capsule are found in some bacterial cells. This additional outer covering protects the cell when it is engulfed by other organisms. Assist in retaining moisture, yes, and helps the cell adhere to surface and nutrients. So, cell wall. Cell wall is an outer covering that protects the bacterial cell, and rigid exterior cell that defines the shape of bacterial cells is chemically complex, providing the rigidity, the strength and protection. The main constituent of most bacterial cells walls is a complex. The cytoplasm. Okay, cytoplasm. Cytoplasm is gel-like substance composed mainly of water. Okay, that also contains enzymes, salts, cell components, and various organic molecules. That is the cytoplasm. Okay. The villi or pilus sigilla is a kind of hair-like structures of the surface of the cell that attach to other bacterial cells. Yeah, attached to other bacterial cells. Shorter than villi called the fimbriae helps bacteria attach to surface and most often observe and gram-negative bacteria. They are composed of polymerized protein molecules called what you call that the pillin. Okay, Pili are much thinner than flagella, have rigid structures and were not associated with motility. The flagella are long whip-like uh, protozoans that aid in a circular locomotion. The bacterial flagella are about 10 to 20 nm thick, so too thin to be seen in the component like microscope. It's too thin. While the ribosomes are cell structures responsible for the protein production. The plasmids are the gene-carrying circular DNA structures that are not involved in reproduction. The nucleoid region or an area of the cytoplasm that contains a single bacterial DNA molecule. The prokaryotic cells are organelles found in eukaryotic cells, such as mitochondria, endoplasmic reticulum, and Golgi complex. Eukaryotic organelles are thought to have evolved from prokaryotic cells, living in endosymbiotic relationship with one another, like like um like plant cells. Bacteria have a cell wall. Some bacteria also have a polysaccharide capsule layer surrounding the cell wall. This is the layer where bacteria produce the biofilm, a slimy substance that helps the bacterial colonize and adhere the surface and each other protection against the antibiotics, chemicals, and other hazardous substances. Okay. And that's the prokaryotic cell and their structures. So we're done to tell you the eukaryotic and prokaryotic cell structures. So now I'm going to tackle their different, the prokaryotic versus the eukaryotic. So let's start. The eukaryotic cells membrane bond in organelles, just like I said earlier. So including a nucleus, and eukaryotic can be single cell or multi-celled, such as plants, fungi, and insects. Contain many membrane includes large complex organelles in the cytoplasm, whereas a prokaryotic cells do not contain these membrane bond organelles. Just like I said in the first video, the eukaryotic cells earlier. So bacteria are an examples of prokaryotes. Prokaryotic cells do not contain the nucleus or any other membrane bound in organelle. Eukaryotic cells are divided into plants and animal types. Animal cells do not usually consisting of cellulose. Prokaryotic cells have a complex cell wall consisting of proteins, lipids, and polysaccharides. Eukaryotic cells contains the membranous structures such as the endoplasmic reticulum and the Golgi complex, and many membrane bound in organelles such as mitochondria and platelets. Prokaryotic cells possess no membrane other than the cell membrane that includes the cytoplasm. The prokaryotic are organisms made of cells that lack cell nucleus or any membrane encased organelles. While the eukaryotic are organisms made of cells that possess a membrane-bound nucleus that holds genetic material, just like I said earlier, as a membrane-bound organelles. So, the eukaryotic cells usually larger than the prokaryotic cells, and they are found mainly in multicellular organisms with eukaryotic cells are celled eukaryotes, and they range from fungi to people. Eukaryotic cells also contain other organelles besides the nucleus. Like a prokaryotic cell. A eukaryotic cell has a plasma membrane, cytoplasm, and ribosomes. But eukaryotic cell is typically larger than the prokaryotic cells. Has a true nucleus, meaning its DNA is surrounded by a membrane, and has other membrane bound and organelles that allow for compartmentalization of functions. But in the prokaryotic and eukaryotic, there is also have a similarities. In the cell membrane, okay, both eukaryotic and prokaryotic cells bear a lipid layer, which is an arrangement of phospholipids, proteins, and that act as a selective barrier between the external and internal environment of the cell. By their genetic material, the eukaryotic and prokaryotic cells both use deoxyribonucleic acid (DNA) as the basis for their genetic information. This genetic material is needed to regulate and inform cell function through the creation of a RNA by transcription, followed by the generation of protein through the translations. The ribosomes facilitate the RNA translation and the creation of protein, which is essential to the functioning of both eukaryotic and prokaryotic cells. The cytoplasm. Okay. The cytoplasm is the medium in which the biochemical reaction of the cell take place, of which the primary component is cytosol. In the eukaryotic cells, the cytoplasm comprises everything between the plasma membrane And the nuclear envelope include the organelles. Yes. The material within the nucleus is termed the nucleoplasm. Is in prokaryotes, the cytoplasm encompasses within everything within the plasma membrane, including the cytoskeleton and the genetic material. Now, we're already done tackling their difference of eukaryotic and prokaryotic cells. So, the reproductions, we're going to tackle now the reproduction of organisms and their cells. So the reproduction are referring to the manner in which organisms reproduce, while the cell reproduction is referring to the process by which an individual cell reproduces. Eukaryotic protozoa, fungi, algae undergo cell division called mitosis in order to reproduce. So, how do mitosis work, and how does a eukaryotic cells experience duplication and reproduction? Inside the eukaryotic nucleus is a nucleolus, where mass of DNA or genetic material resides and chromatin spread. This event is called interphase, as the chromatin material condenses and experience of destructive set of chromosomes are observed. DNA is a duplication course which will create set of chromatids, or could also be called sister chromatids. So this period is called early phosphate. The nucleus disappears, the centrioles moves towards the opposite end poles and began to form the spindle fibers of microtubules, which is known as a late phosphate. Next to this is a metaphase, where spindle fibers continue to strive into the opposite poles, and the chromatids line up in order in the spindle cleater. So, pairs of chromatids are attached to the kinetochore. In the kinetochore is the centromere region. DNA is the most chromosomes arrive at the end poles. Thus, the developing new nuclei forming two nuclei. Also, as the point division of cytoplasm begins. The cytokinesis is the process that follows mitosis. The microfilaments constrict the cell at the spindle equator, separating the cytoplasm into the two daughter cells. Each newly developed daughter cells tends to have its own cell nucleus as a nuclei envelope is being synthesized. So this is how mitosis work in a eukaryotic cell. Now let's move on forward to the next process that takes place in order for the prokaryotic cells to divide. Okay? The prokaryotic bacteria or binary fission is the process of how prokaryotic cells divide. Even before the process of binary fission begins, there is an advanced duplication of bacteria. Bacteria chromosomes or DNA grows in the cell membrane. It separates. It it will form two new chromosomes in the nucleoid region, and again these two will undergo separation of new cross walls. If completed, two new daughter cells are being created. So this is how binary fission works. And so this is how binary fission works. To adopt some information, a eukaryotic and prokaryotic cell or microorganism is general experience cell division at the different times intervals. The varying times used in cell division is termed as a generation time. The Eukaryotic cells reproduce by a process known as a binary fission, just like I said earlier, in which one cells, the parent cells, splits in half to become two daughter cells before a prokaryotic cells can divide in half. Its chromosomes must be duplicated so that each daughter cells will possess the same genetic information as the parent cells. The time it takes from binary fission to occur is called the generation time. The generation time varies from one bacterial species to another and also depends on the growth condition. So taxonomy. Taxonomy, the science of classification of living organisms. Taxonomy consists of three separate but interrelated areas of classification: the nomenclature and their identifications. Classification is the agreement of organisms into the taxonomic groups on the basis of similarities or relationships rather. Taxa include kingdom or domain, division of phyla, classes or orders, families and species that closely related on organism placed into the same taxon. Nomenclature is the assignments of names to the various taxa. Identification is the process of determining whether an isolate belongs to one of the established named taxa or represents a previously identified species. When attempting to identify an organism that has been dissolved from a clinical specimen, laboratory or technologists are very much like detectives that they gather clues about the organisms in most cases, and clues that have been gathered will match the characteristics of an established species. And again, my name is Jeffy Santos, and thank you for watching. See you on my next topic. Bye.